So a quick reminder that cardiac muscle cells are unique. They're not fused together by tight junctions. Rather, you've got gap junctions at intercalated discs. Remember also that cardiac muscle cells are very branched, and this allows fast signal propagation in 3 different directions. And that means that the whole of the atria, for example, can contract as one big unit, as well as individual cells being able to undergo muscular contraction. Now, what's also special is the idea that the heart is myogenic. It can beat by itself in the sense it doesn't need to have a direct nervous input. So the direct nervous input is there. That comes from the autonomic nervous system that can speed the heart up. It can slow the heart down. But there is a general intrinsic heart rate or autorhythmic cells within the heart, the pacemaker of the heart, which sets that pace. And that's what we're going to look at. We're going to look at how the pacemaker works, how the pacemaker causes atrial contraction or atrial systole, and then atrial systole is followed by ventricular systole, and then finally diastole. So there's 3 stages of a cycle of events that take place in the beating of the heart or the cardiac cycle. So atrial contraction or atrial systole are done by the cardiac cells in the atria. Now, as the atria get excited, it more or less beats as one whole. Remember that the atrial walls are thin, so this is relatively straightforward. Then what happens is that there is a deliberate pause, and there's a pause because the atria and the ventricle are separated by a sheath of connective tissue. But then what we have is events, and I'll explain those to you in a bit of detail now, ventricular systole or ventricular contraction. And then you've got a period of time where the heart is in a relaxed mode. So the relaxed cells or diastole is followed. Now, this comes from BioNinja, and BioNinja covers this very nicely. And you can see the events taking place as we go across the bottom. But I want to draw your attention to this patch of tissue up in the right atrium. This is the pacemaker, and the pacemaker is leaky to ions. And because it's leaky to ions, it suddenly becomes electrically excited. And when it becomes electrically excited, that signal is then transmitted across the atria, the left and the right atria, more or less as one whole. So what I've done here, which I hope is helpful, is I've taken you through the series of events and pointing out in bright green there the big signpost for you to take away. So there are a region called the sinoatrial node. The sinoatrial node is the same thing as the pacemaker. It's made up of specialized cells called cardiomyocyte cells. And the pacemaker, that patch that I just showed you in the right atrium, is what begins the process of atrial systole. The electrically excited region here is then propagated because of the branching of the cells and because of the loose junctions, the intercalated disc. So you get fast signal propagation, then you get atrial systole, the contraction of the muscle cell. So you've got a wave of excitation and then a wave of contraction which follows that particular event. Then you've got the atrioventricular junction, and the atrioventricular junction is a second patch of tissue or a nodal tissue which becomes excited, and I'll point to you where that is next. So here we've got the sinoatrial node, the pacemaker. We've got this then here, which is at the junction. If we go back to this slide, it's at the junction here. So the atria and ventricle are separated along this sort of line here. You've got tough connective tissue, so that wave of excitation as it passes along can't go further than this point. But this patch of tissue here is excited, and that's referred to as the atrioventricular node. So you get excitation of the pacemaker, excitation spreading across the atria, and then it reaches this patch, which is called the atrioventricular node. And this becomes excited, and it's going to propagate at a much slower speed, an impulse which goes down the middle of the septum through here, and then it starts to branch upwards from the apex upwards, and these blue endings here are going to excite individual muscle cells, and this is a type of nerve called Purkinje fibers. So we've got the pacemaker contracting or initiating the contraction of the atria. That hits the atrioventricular node. When that is excited, it shoots a signal down through the septum, no innervation, and then it branches upwards through these Purkinje fibers, and those Purkinje fibers are then going to cause the heart to contract from the bottom of the heart or the apex upwards. So here from BioNinja, again, you can see the series of events, and we're interested now in what's going on the bottom here, the bundle of hairs are these innervations of the tissue of the ventricle. Again, to try and make it a little bit easier, I've put those into prose that you can read and take away, and anything on there that you are unfamiliar with, that you don't understand, you can come back to me. So we've got atrial systole, ventricular systole, and then finally we've got this diastole period. And diastole means that the heart is at rest. Now there's something called a refractory period that you don't need to know much about, but it basically means that even if the heart is excited by electricity, it won't beat. So it undergoes a period where it says, right, I'm shutting off from the world, and then it comes out of that refractory period, and then it can be excited again. Now there's a long refractory period in cardiac muscle compared to skeletal muscle, which means it's forced recovery, and that's the reason why that the heart can carry on beating for a long time and it doesn't become fatigued. So here I've tried to summarize everything that I've just said. What I've included here, which I haven't mentioned yet, is the valves that are involved. So when we get atrial systole, that is the contraction of the atria, we're going to push open the atrioventricular valves, and they are going to be pushed open because the blood pressure inside the atria as a consequence of its contracting is going to increase. And the atrial pressure needs to be bigger than the ventricular pressure, and that's going to move blood from the atria down into the ventricle through these atrioventricular valves. Remember, the atrioventricular valves are your bicuspid and your tricuspid valve. Now, at this point, the semilunar valves are going to be closed, and the artery pressure at this point in time is quite high, higher than the ventricular pressure. So when all of this takes place, what you've got is atrial systole, contraction of the heart, generation of a high blood pressure in the atria, pushing open the valves, and therefore you get this ventricles filling with blood. So all this part in green is all associated with atrial systole. Next, we've got ventricular systole, and this is when the ventricle muscle contracts. It's going to contract from the bottom or the apex of the heart upwards. All of the blood inside the ventricle is therefore going to be under high pressure, and that's going to push upwards against the atrioventricular valves and also the semilunar valves. Now it's going to push the atrioventricular valves closed, and that prevents the backflow of blood, and it's going to push open the semilunar valves because the pressure now in the ventricle is higher than the pressure in the artery, the aorta or the pulmonary artery. That means that blood is going to flow into these arteries, and at the same time what you have is atria refilling with blood because you've got the continuous blood flow from the veins, and the veins here are the vena cava on the right side and the pulmonary veins on the left side. Then following off from here, from ventricular systole, we've got the diastole, and the whole of the heart is undergoing this forced rest. The ventricles stop contracting, so the pressure inside the ventricles fall, and because it falls, the semilunar valves are going to have blood moving backwards against them from the artery. So the semilunar valves are going to snap shut as a consequence of some blood going back, but then that's going to ultimately prevent more blood flowing back into the ventricle. And again, at the same time, the atria are filling up with blood, and the cycle is going to start all over again after that. This is a diagrammatic version of everything that I've just said, which is focused on pressure. And you need to appreciate here that blood will always flow from a high pressure to a low pressure. And what I want you to do is to pause the video in a moment, and I want you to look very carefully at the regions A, B, C, and D. And I want you to think about the difference in the pressure there and the events that might be taking place. And I'll just pause. So think about the differences in the pressure in A. Think about the implication for the valves when that takes place. Same for B, C, and D. Now, I suggest you switch this off, work it out before you carry on playing. Now, this is a bit of a favorite with the examiners, so take care to unpack this and understand it. But if we look at this part of the graph here for A, what you should see is that the pressure in the ventricle is greater than the pressure in the atrium. So that's when you get this snapping closed of the atrioventricular valve, and that causes the sound, the lump sound of the heartbeat. If we look at B, again, you can see the differences in the pressure. The ventricle pressure here is greater than the aorta pressure, and that means that the semilunar valves at this point are going to be open. Here at C, the semilunar valves are snapped shut. The aorta pressure is greater than the ventricle, so blood will start to go back into the ventricle, but it'll push those valves closed, and they remain closed, and that's the dup sound of the heart. And then D, what you've got is atrial pressure, this time being slightly higher than the ventricle pressure, which pushes open those atrioventricular valves. This diagram here is showing you exactly the same idea. And here you can see the lup dup sound, which is associated with the heart sound. So we do need to know, the syllabus statement wants you to know about the sounds produced as those heartbeat, as those valves move. Again, okay, there's a diagram here. You've got the lup and the dup. You must make sure you can appreciate the differences in the pressure and the opening and the closing of the valves which are associated with that. So first sound, this is from BioNinja, are these valves here closing, and then these valves here closing. And this one is asking you to not just talk about the heart and the blood circulating through the heart. This is asking you to look at the actual ECG or EKG. It's exactly the same thing. It's looking at the electrical sequences which take place in the heart. So all of this, and going back to this graph, what we're looking at here are pressure changes, and the pressure changes are associated with the contraction of the heart. Here, when we're looking at the ECG, what we're interested in doing is understanding the electrical activity within the heart. So we've got an electrocardiograph which shows data, and the data is called an electrocardiogram. And the analogy here, again taken from BioNinja, a very nice resource. You've got a telegraph machine. I don't know if you've ever heard of a telegraph, it's quite old, but it's going to send a telegram. So the electrocardiogram is the trace that's produced. And what we're going to see is a P wave, a QRS complex, and a T wave. And again, it's quite a favorite of the exam scheme, and we need to appreciate what each one of these is representing. So here we've got the P wave, that bump there, and this is all about depolarization of the atria. Now, depolarization is electrical excitation across the cell membrane of those atrial cells. So as the pacemaker operates, it's going to send out a signal, and that signal is propagated as it hits the surface of individual cardiac muscle cells. It's going to cause them to depolarize. It's going to cause ions to move across that membrane, which has got an associated electrical flux with it, and that's what we're seeing here. Then we've got the QRS complex, that's here. And what this is representing is the depolarization of the ventricle. So as the ventricle contracts, because of the actions that have come through the bundle of His, we've got this trace being recorded. And then we've got the T wave, which is the other bump here that we can see. And this is repolarization of the ventricle. So here, what happens is actually sodium is moving across the membrane and into the cells. And here, we're moving it back out again. So depolarization and repolarization are electrical events because of the movement of ions. Now, you don't have to know it in so much detail, but you do need to be able to recognize that this happens, then this happens, and then this happens. And because you've got one heart rate following the next, then you should get, on a normal electrocardiogram, a trace where this is repeated exactly as it is here over and over again. What you need to appreciate is that by looking at an electrocardiogram, you can identify certain heart conditions. So tachycardia is when your heart is going too fast. Brachycardia is when you're going too slow. Arrhythmias are irregular, where it jumps all over the place. This one is particularly common as you get older. And fibrillations are really dangerous, and that's when the heart is contracting in an irregular way. These are examples of the traces that we should see in a normal trace, and then you've got too fast, too slow. This one is the fibrillation one, is all over the place, and you need to be able to recognize these traces. Now, this last little bit we're going to move on to next time, so I'm going to pause here and allow you to look through those slides, but we need to think about how the heart rate changes, but remember that the initiation of the heart is done by the pacemaker. We'll come back to this next time.